Yeah. Can you give me permission for slides?
Yep. I am going to the actual recording of the video will start when you start talking. Can you try to share and then I can give you
Mhm.
permission?
Yeah.
There you go. Okay, and you can introduce yourself and consider it recording now for all of the people on the phone.
Okay. Um, Great. So I'm going to go into presenter mode, I hope. Uh, can you see the large screen or the small screen?
I can see both. The small screen is too big.
So I can swap it. Does this help?
That's great.
Okay. That's what I need to know. Let's see. Okay. So my name is Larry Ryder, and I'm a professor in the Department of Physiology at Tulane University. I was previously a professor for about 20 years at the University of Tennessee Health Science Center in Memphis, Tennessee. And there we developed a program to study rare diseases using pulp and stem cells that live in the pulp of teeth. Specifically, we collect baby teeth from a variety of rare disorders and would love to start collecting some USP7 and some SLC6A1. And today I was going to go over sort of the process of how we do it and what kind of things we've done in the past. And then at the end, there's only a few slides, but at the end, I could take questions about it, and I'm happy to do that and show you the code to to fill out the consent form. Okay, let's see if I can go forward. No, I cannot. All right. So, what are dental pulp stem cells? So, dental pulp stem cells live deep in the pulp of the tooth, and these stem cells um, they grow immediately, unlike iPSCs, which you make from skin biopsies. These you can just put in a dish and grow. They can be easily differentiated into these cultures that we call cortical light cultures, because a lot of what we do is related to neurons. And they've been differentiated into lots of different cell types. So you can make chondrocytes, adipocytes, glia, and at the end, I'll show you can even make kidneys out of dental pulp stem cells, which is really cool. We can get a lot more biological samples from rare disorder this way. One of the diseases we study is called CCHS, and it is an extremely rare disorder. And the only way we've been able to study samples was by collaborating with the one clinician who sees pretty much all of the 1,500 kids that have this disease. So we're pretty familiar with the process, and it's been very useful for rare diseases. So I won't go into too many details about epigenetics and other things, but these cells, they actually resemble embryonic stem cells more than they do iPSCs at many levels. It makes them a better candidate for using in the lab to reflect what's really going on in a particular disease. So how do we do all this? Well, really, it's come down now to a very simple collection kit. We used to send out the consent form, but now we've put the consent form online on a Qualtrics system. So it allows us to pretty much you go to that link, you fill out the consent, you upload your genetics report, and then we send you a box, and the box has a tube of media. And this media is just basic cell culture media with some antibiotics. When your child's tooth falls out, you put it in that tube and you put it back in the box with a return FedEx on it. So it's got all that stuff already. You don't have to pay anything. You just put it in the box, send it to us. Hopefully, that you get the tooth in the media within about 20 or 30 minutes of the time it came out of the mouth and then get it to us within 48 to 72 hours. So hopefully it doesn't come out like Friday night, but If it does, you can put it in the media and it will keep until Monday and you can send us the kit Monday. You can put it in the fridge actually. So on the right side, you'll see we've been doing this for about 15 years and we have over 200 dental pulp stem cell lines from 14 different syndromes, with a lot of emphasis on syndromes related to 15q, Angelman, Prader-Willi, and duplication 15q. We have a lot of those lines. But we've also got a lot of other types of disorders like Wagner syndrome, which affects the kidneys. And I'll show you at the end, I've been working with somebody to actually make kidneys from dental pulp stem cells so that we can study this disease. So what do they look like? So when we grow the cells in a dish and then stain them, in this case with beta tubulin, you see that they look very much like what we call fibroblasts. They just look like these flat cells. Using the differentiation process that we use, which does not involve any kind of virus or anything, we just coax them into these neuronal lineages. They end up looking very much like neurons. Not all the cells in the dish are neurons, but a lot of them are. And they have the right markers when they transition to be either, in this case, GFAP, so there are glial cells in there, but there are also neurons in there that have both signals for MAP2 and GABA A. This is the transition from nothing in the DPSEs to very high expression in the neurons that tells us that we're making neuronal cultures. And finally, for those interested, these neurons actually can fire currents, sodium, potassium, calcium currents. We've measured all those, we and other labs. And we're actually going to start using them in a system called an MEA system, which will allow us to monitor their neuronal activity just by plating them on this system. So I'm going to talk a little bit about two projects that relate to Prader-Willi syndrome, just so you understand generally what we can do with these cultures. And I'm really going to summarize both those projects in 2 slides each, so it won't be very, very detailed. This one's interesting because in the case of Prader-Willi uniparental disomy, those individuals have a higher incidence of ASD than other individuals, so a higher incidence of autism. So we were interested in seeing if there were any molecular changes that were also coming along with that diagnosis of ASD. And when we looked at the kids that had PWS plus ASD versus PWS UPD minus ASD, we found that there were these inclusions in the neurons. And really all I'm showing you here is, we're looking at a marker for mitochondria, and that marker says that these mitochondria are basically clumping together. And if you look above it in the kids that had PWS UPD minus ASD, there's a nice pattern of expression throughout the neuron. So this is an indicator that something is wrong with the mitochondria in these neurons. And in fact, when we quantify it, you can actually see that there's a decrease in the volume of staining for that marker of mitochondria. So now we're in the process of figuring out exactly what's wrong with the mitochondria. But we have a clear indicator that when these children have autism and PWS, UPD, they have this mitochondrial inclusion. When they don't have ASD, they match the other deletion cases in the controls. Their mitochondria don't appear to have this defect. So here's another story, another thing that we can do. We can take, in this case, a virus that has a reporter for circadian rhythm called PER2 that's linked to luciferase, which is a protein that lights up, basically. So when HER2 expression is on, this luciferase lights up the cells, and we can actually measure that and we used to use a different method, thing called a Lumicycle, and I'll show you that data. We can measure that light over time. And what's interesting about it is, if you look at multiple control individuals, so we made neurons from control teeth, they have this very nice circadian pattern over time. It goes up, it goes down. This is what controls your sleep habits, right? Is your endogenous circadian rhythm. But when we looked at Prader-Willi lines, we found that they had a shift in their period. So they actually had a shorter period length. Now, what you may or may not know about Prader-Willi syndrome is those children also have chronic daytime sleepiness. Why do they have daytime sleepiness? Well, one of the reasons might be because their circadian rhythm isn't right. So we're trying now to figure out why that is. There are some clues um, from the circadian rhythm literature that one of the proteins involved in Prader-Willi syndrome, MAGEL2, you'll notice another protein that some of you know here, USP7. So MAGEL2 interacts with USP7, and USP7 regulates one of the clock genes called CRY1, which is essential for maintaining this circadian pattern in the cell. So we're in the process of dissecting this and figuring out how MAGEL2 mutations, which cause another disease called Schaaf-Yang syndrome, or MAGEL2 deletion in Prader-Willi syndrome, how those affect the circadian rhythm, and is that solely through USP7 interaction, and do they regulate CRY1? So we're interested in all those things. And hopefully we have an NIH grant that will get reviewed someday, whenever that happens, that's on this topic. Okay, and one last major science slide, and that is we recently started making what we think are brain organoids from dental pulp stem cells. And we're not exactly sure what's going on in terms of the structure of the organoid, but we've been able to do the MEA system on them and show that they have patterns that are indicative of neuronal firing, and that in the case of, in this case, in Dravet syndrome, which is a severe epilepsy syndrome, we saw uncontrolled firing in those particular organoids compared to the control organoids. And then on the bright side, I'm going to show you something even crazier. I have a collaborator in the department who knows how to make kidney organoids. That's what this picture is. And you'll have to trust me that he says these are primitive ducts, that this is sort of the formation of the ductwork that happens in your kidney to filter toxins out of your blood, right? That's how your kidneys work. So he has done this before, and this is a very immature organoid, but he's successfully been able to make this particular organoid from dental pulp stem cells, and we're hoping to do that from another syndrome called Wagner syndrome that is associated with kidney tumors. And we're going to see if we can actually look at that process of how those tumors form by using these organoids. So a couple just general conclusions. Dental pulp stem cells are a really viable alternative to iPSCs. Some of you may have even donated skin biopsies before to make iPSCs. You don't need to do that to make dental pulp stem cells. You just need to have baby teeth that come out, which are going to come out anyway. We've done a lot of cellular and molecular studies using our DPSCs, and we figured out some very interesting things, mostly about Prader-Willi syndrome, a little bit of Schaaf-Yang. We're continuing to do that work now. And we've found some very interesting things related to autism and circadian rhythm just using our dental pulp stem cells. They've really proven very useful for modeling the methylome and endocytic recycling. And even recently, we did some work with a collaborator on mechanosensitive ion channels, which is kind of cool, I think. So we're just scratching the surface here. We're open to new syndromes. That's why I'm speaking to you guys today, because we're happy to collect both SLC6A1 and USP7 teeth and try and make some DPSC lines that we can then use for future studies of these particular syndromes. And this just gives you an idea of the kind of stuff that we've published. Quite a few papers in the last 10 years, and a picture of I'd say about half my current lab. Even though this was back in 2024, I've grown quite a bit. But some of you may have met Taylor at the USP7 meeting. Tyler is a postdoc in my lab. Taylor is a grad student, and Tyler has been working the most really with dental pulp stem cells in Prader-Willi syndrome. And Ben is a fly guy in the lab. He works on a Drosophila project. So the last slide is um, the QR code that you can scan and go directly to the Qualtrics consent form. But when you do that, you need to have a PDF copy of your genetics report or your child's genetics report so you can upload that at the end. So we need that, and there's some questions you have to answer. And then at that point, sometime later, you'll be sent another survey that's an autism survey that we use on everybody that's online as well, called the SCQ, Social Communication Questionnaire. And that's it. Now I'm really, really happy to take questions, actually. Been talking too long.
Well, the first question would be, you mentioned that you can order kits now. How far in advance should people order and how long are they good?
So I would say that tube of media is probably good if you put it in the fridge for at least a year. So you kind of want to get involved in this when your kid is starting to lose teeth or has wiggly teeth, right? You don't want to get the kit when they're like 2 years old, right? And it's going to be another 5 years before they lose a tooth, That's a little too soon.
Do you need siblings or any controls?
I would love some sibling controls, and that is an option as well. You'll have to go through the consent form again to do that, but there is an option for if you don't have a syndrome, and I believe it's been set up correctly for that so that you can go through the process with a different kid that is a sibling. Yeah.
Okay, and will families receive any follow-up if their cells are made into organoids?
No, everything is really one direction here. So it all goes into a repository that we have.
Oh,
So
cool.
when it gets in the repository, it becomes de-identified, and it's just a cell line that's associated with particular disease, particular mutation. And that also allows us to share these lines with labs and do collaborative studies. The cells are not immortalized yet. Um, so unlike iPSCs, DPSCs will undergo cellular senescence at about passage 14, 13 or 14. So what we do, we have a way to get around that now, and we're in the process of doing that. But for the most part, we can only really share them with other labs when we have a direct collaboration because they're a limited resource because they don't last forever. But what they will hear from us is if we can't grow a tooth, we may contact them again about with a tube, and send them another tube of media to get the next tooth, right? Because sometimes they just don't grow, or they grow and they're contaminated. That happens, because it does come from your mouth. So
Yeah, that definitely makes sense. And for the repository, who can access that repository?
I mean, really, it's just our in-house repository. Um, so like the only people that actually access it are me and Tyler, and I have a genetics master's student who's been organizing it and trying to help us with shipping kits and all that kind of stuff.
But technically, the repository, if you grow organoids, would other scientists be able to access it? I saw some folks from Italy join the call that are scientists, and they actually would be interested, I would guess, in accessing the organoids.
So we don't have any organoids. We only have dental pulp stem cells in the repository. And the one organoid project I'm working on, well, there's two. One is the kidney project, and that's just new. That's been like 6 months of just trying to do it, and we actually got it done. And the other, the brain organoids, my postdoc started trying to make. And so all of that is so, you know, brand new that we don't have anything to share. We don't even have anything we've published or, it's very new. Now, as far as getting dental pulp stem cells, um, it's funny you mentioned Italy because I have a postdoc coming from Italy in January.
Really?
Yeah, it's a really good story. I don't know who's on the call. Maybe it's her boss. So I went to the European Society for Human Genetics meeting last year, and someone presented and they kept talking about our publications, and they were working on a disease that we actually work on, this rare disease CCHS. And they followed our protocol and they made dental pulp stem cell neurons and they did single cell analysis. And so I talked to her later and she's interested in coming to my lab. So she's going to be coming in January. But in the meantime, I sent them other CCHS lines so they could finish doing the single cell work. So I did send dental pulp stem cells to Italy. Now, the problem with that is they got stuck over the weekend in customs, but we tried again. I think she finally got the lines. In fact, I have to follow up on that because they just got stuck for like a week and a half in customs, and thawed out. But it's tricky to send anything internationally. Let's just put it that way.
It terrifies me because this is such precious material and customs doesn't understand.
No, they don't. They don't care. But I mean, we have copies, so it wasn't the only line, but it just is unfortunate that it's really hard to get through that barrier to get other researchers the resource.
Yeah.
It
We have a question how long this study is?
it's been ongoing forever. I mean, I started this, like I said, 15 years ago, and right now it's just open and I renew it every year. So I don't plan on stopping because I work on so many different syndromes. Um, yeah, like it depends on what happens when we ask your communities for teeth, right? So the only way I would slow down on collection is if we just get way too many, right? And that's happened before. Like we were collecting some tuberous sclerosis lines and it's just way too common. It turned out, we had an onslaught of tuberous sclerosis and you know, we didn't need that. We weren't even working on it really. We were working on it with a collaborator who ended up not using those lines. So, I still have them, but I had way too many and it cost money to make the lines. So we had to stop doing that. But um, I can't imagine that either USP7 or SLC6A1 would be so incredibly common that I got overloaded with teeth. Um, so let's try it first. you know see what happens.
Johanna's son is I I would say about 9.
Yeah, it's a good age.
Yeah, and I think that he's an excellent person to consider to study.
Yeah, I mean, fill out the consent and I will get an email and then we'll put the kit together. And the only thing that's critical for SLC especially is that I see that genetics report because everybody in the repository, I need to know what they actually have. And for
Yeah.
SLC, like for some of the syndromes, everybody's got the same thing. For some of the disorders we collect. But for SLC, there's so many different mutations. I need to know who's got what.
Okay. Makes sense. Any other questions? I think we're good.
I don't bite and I'm here, so you can ask me whatever you like.
Well, thank you so much. And I will follow up with any questions I receive after the fact. I'll put my email in the chat.
And please remember, if you're going to scan that and go to the survey, which is a Qualtrics survey, that's the consent form, at the very end, it'll ask for the genetics report. So don't start doing it until you have that report handy. You know, you have a PDF of it or something.
Absolutely. I will remind everybody.
And there's a link right there too, which is the same thing as that QR code that goes to the link.
Okay, do you have the link handy to put in the chat?
It's right there. Do you want me to
Oh,
put it in the chat? I think I could do it. I'd have to like, oop.
don't worry about it. That is pretty self-explanatory.
But it might be easier for people if I copied and pasted it in the chat, right?
Okay.
Let me see if I can do that. I will try. Yeah, I can do that. Is the chat. Now I can't find the chat. Oh, there it is. Okay. Admin restrictions. Did it go through?
Yep.
Okay. It said
Okay.
something about admin restrictions. I didn't know if you couldn't put links in there or something.
You are good.
Excellent. Okay, guys. I appreciate the chance to talk about this.
Well, we appreciate you and everybody have a great day.
Okay. Bye.
Bye.
Bye. Thank you.